in the United States, we can't even get genetically modified food without a big controversy. Well, we have some really interesting news out of the ESC meeting here. It's phase 2 clinical trial results of a first-in-class single-dose gene therapy, and it's a treatment for patients with reduced left ventricular ejection fraction and, of course, heart failure. So I am with the Renova Therapeutics CEO, Dr. Jack Reich. Thank you very much, first off. And we can't, in the United States, it's like gene-modified this or whatever. It's controversial, and you're taking a treatment, and it's a one-time treatment, too. Go back to the beginning before we talk about the phase 2 and tell me about this RT100.
Sure. First off, thanks for having me here today. So this is a treatment that we've developed, which is based on a human adenovirus, common cold virus. It's been modified so it can't reproduce, so it doesn't cause colds. And in that space, we put the human gene, AC6, which is one of the main factors in the creation of energy, contraction, and the creation of energy in heart muscle cells. And what we found early on, and the reason we pursued this was that Dr. Kirk Hammond, who's our scientific founder, is a professor of medicine at UCSD, cardiologist, he discovered that patients with heart failure all had downregulation of AC6 in heart muscle cells. So he reasoned that if he could restore AC6 levels to normal, that he might be able to restore heart function to normal, and that's essentially what's happened.
So what did you see in animal data first?
Sure. So there was an extensive preclinical development, actually the largest I've ever seen in biotechnology. So about 40 published works, peer-reviewed, placebo-controlled, and what Kirk was able to show in various models of different types of heart failure, different animal models, was that essentially a single dose of this gene therapy could restore heart function to normal. Now this is extraordinary, both in low ejection fraction and normal ejection fraction heart failure. And so based on that and formal toxicology, that program was supported by the National Institutes of Health, so they played a key role in this. And the product was atoxic, which was consistent with the last product we developed in the last company that we developed. So we knew the safety was great. And that followed through in the animal work. As a result of that, the NIH awarded Kirk Hammond a grant to do a clinical trial of this therapy in patients. And so they designed the study together and then filed an IND and so on. And then a little while after that, Renova Therapeutics was formed to develop the product beyond that. And so one of the first things I did as the CEO was I felt that it would be best if we could partner with the NIH. And there were regulations that allowed that. My background is in regulatory affairs. And so I pursued a public-private partnership, and that was put together over about a year and a half. And so we signed a public-private partnership with the NIH. That allowed the trial to go along smoothly and then be completed.
So what's being presented here?
Well, the results of the trial. So let's begin with the safety, which is obviously very important in an initial trial. The trial design was a classic dose-response study, what you would expect in phase 2. And what we found was, overall, that the safety was extraordinary in the study, meaning that there were nothing of concern in the safety in these 56 patients, 42 of whom received the gene therapy. So essentially, the placebo patients looked just like the gene therapy patients. And that was the case in our last gene therapy development. So we really kind of expected that, and that's what we saw. It's very important. Now, many people, when you would talk about a gene therapy like this, would be concerned about flogging the heart, right? Causing the heart to beat faster and so on, which would actually bring on earlier death in patients like this. So our gene therapy does not work in that way at all. So there were no increases in heart rate, no increases in blood pressure, and just as importantly, it produced no arrhythmias. So the safety profile is just what you would want for a single-dose gene therapy. Now then you look at the efficacy, and the efficacy likewise. So we were looking for dose response. The 3 lowest doses, which we did not actually expect to have efficacy, if you will, actually had signs of efficacy. In the first dose group, for example, at 12 weeks, we saw a statistically significant improvement in ejection fraction. That's a signal, just at the very first dose. But when you get to the two highest doses, which we had previously had history with both in animals and humans, in fact, we saw what we expected to see, which was clear signals of efficacy, and I can go into those for you, and actually a building of efficacy from dose 4 to dose 5. So exactly what you would expect. And so those two doses will go forward in further clinical trials.
So what were the efficacious signs you talk about?
Sure. So one was ejection fraction. Now, I would think that all cardiologists would recognize if you're talking about heart function, that would be a good thing to improve ejection fraction, right?
True.
Okay, and that's what happened. So what you see here in these particular patients, and we focused on the two highest dose groups, was that, for example, in the highest dose group, at 12 weeks, over a baseline of around 30 units for ejection fraction. So these people obviously in the study were sick. They were not healthy volunteers. These people had low ejection fraction heart failure. The improvement over 12 weeks from baseline of 30 was over 8 points. Now this is serious business, right? Because as you're probably well aware, you can see changes or improvements of 5 points or less just from variation day to day and so on. So those are sort of not proof, if you will. But when you get beyond that, now you're talking about something really material. And so what we saw, for example, in the top two dose groups was about 40% of the patients had improvements that were greater than 5 points and as many as 24 points. So many of these patients, for example, have gone from being at 30 to being in the high 40s or 50s. That, as you know, you can't make, that's not a placebo response.
Right, exactly.
Okay. So, and obviously, none of the placebo patients made it above 5 points. So that was statistically different. Clearly, statistically significant. Then likewise, we did something that's hardly ever done in clinical trials, was to look at DPDT. Now, that's done in animals, of course.
Right.
But very rarely, if ever, is it done in a medical trial like this. So in fact, what we saw there was a statistically significant improvement in the gene therapy treated patients in negative DPDT, just what we saw in animals. So the relaxation of the heart is much better in the gene therapy treated patients than in the placebo patients, statistically significantly so. So you've got the two main measures of heart function, both improved. We saw improvement as well in time on a treadmill, but as has been published before, patients in these kind of trials, there's a big placebo response. So the placebos did well, the gene therapy did a little better, but not statistically so because of that placebo effect. But the real importance of this is how did it translate into morbidity and mortality, which is what this is all about. And in fact, here we saw really striking results. So for example, the study was a one-year study. Patients get treated and followed for a year. And what we saw was, in terms of episodes of hospitalization for heart failure, the gene therapy treated patients as a group, all of them, showed a 67% decrease compared to placebo in episodes of hospitalization for heart failure. And likewise, in mortality, the placebo group in the first year showed a 7.1% mortality in heart failure, for heart failure, okay? Which is consistent with in-hospital major medical center trials. Now, if you go to outpatient trials, you can see death rates as high as 26, 29%. But in inpatient hospital trials like this, where they're carefully followed, given very good treatment, 7% to 10% is kind of what you see in a year. So we saw 7.1%. But in the gene therapy treated patients, 42 of them, there were no deaths from heart failure.
Wow.
Now, we've got to do bigger trials,
Right.
but this is a heck of a good place to start.
Yes. So what is up next?
Well, next is meeting with health authorities, as you might imagine. And that's my background. I've got a lot of experience, registered products here in the United States in my former life. So we'll work closely with the health authorities to look at pivotal trials that will go forward, how they should be designed, what numbers. I mean, I pretty well know this stuff in general, but you have to have specific discussions. However, having said that, this is a major cause of death that we're talking about here. And with results like this, I believe and I would forecast that the health authorities will be very interested to work closely with us to help us to design those studies going forward. And if the results look anything like what we've seen initially here, I would say there's a very good chance that there might be a registered medicine come out of this at some point.
Now, the question that I have for you is medicine and industry, it's effective treatment, but it's usually long-term treatment. This is a one-time treatment.
Yes.
How do you make a profit off of that? I mean, that's basic, but I mean, how do you, when you've got just one treatment and the patient is in good shape?
Oh, well.
I mean, it just seems a little odd when you look at healthcare in general.
Well, it's not the way we think about things because we think about things from drugs. And drugs treat typically symptoms of disease.
Right.
And what they do in this case is they lower the body's demand for the heart to function. The gene therapy that we're developing actually improves the heart function. Okay, so that can have all kinds of ramifications. Now, having said that, we understand that others who have developed medicines like for cancer and things like that, biologics, first they charge a lot of money, hundreds of thousands of dollars. These things generally will increase life by 3 to 6 months. Okay, what we're looking at here is a different order of magnitude.
Absolutely.
Okay. And so first of all, we will seek a large global partner. So that's very important. Pricing decisions will be made with that partner, but we started early. So a year and a half ago, we began a program with the RAND Corporation, which is a recognized expert in the field. They write what they want to write. So they're working with us directly to help us to determine the cost that will be saved by this medicine if it reaches the market. And therefore, it will dictate to us what we should charge. Now, we want to save money for the healthcare system. We're not trying to take up all the money that is being saved. That's not what we're going to do. And also, we're looking at creative ways for payers to pay over a period of time rather than up front, which is a big burden on the healthcare system. So this is something we're very aware of, and we intend to do this not just with our first therapy here, but also we're developing a gene therapy for type 2 diabetes, which is a simple IV injection one time. So that's where we're headed.
I would like to invite you back whenever you want.
Okay.
Give me a call, give me an email, and we'll set you up again, because this is a fascinating topic, to say the least.
Thank you very much. Enjoyed talking.
Oh, you're welcome. And for CardioSource World News, we have all kinds of things happening here at ESC. Please check the magazines and, of course, online, where I'm Executive Editor Rick McGuire.